Now, we first contacted it when we were talking about blood type. We talked about A antigens and B antigens and D antigens. So, an antigen is a substance which stimulates an immune response, and the immune response is the body's reaction to an antigen. Does that clarify? So, using the definition of one to provide a definition of the other. But an antigen is going to trigger the B cell or T cell reaction. So here's another view of an antigen. And an antigen has multiple surfaces, polysaccharides, lipids, amino acid sequences. And so a T cell receptor or a B cell receptor, similar to an antibody, does not recognize the entire antigen, but recognizes a small segment of that antigen, called the antigenic determinant or epitope. So in research, a scientist will use antibodies to help them isolate certain molecules. And so let's say that there's a protein in the blood. I'm trying to isolate out one of the complement proteins. And that complement protein could be antigenic, but it's going to have multiple surfaces. So a particular lymphocyte might make an antibody against this particular epitope, but a different lymphocyte would make an antibody against this one. And those are then called polyclonal antibodies. So they're each made by different lymphocytes, because the lymphocytes' antibodies are going to recognize always the same thing. But they all recognize the same antigen. Sometimes, for more specificity, scientists want monoclonal antibodies. Currently, early pregnancy tests use monoclonal antibodies. They're more specific, all right, and they can use smaller amounts of the human chorionic gonadotropin that they react to. So just a little bit of the research part of how they develop that, using the different aspects of an epitope. Mice are often they're a chief experimental animal, and they would be given an injection of whatever the whole antigen is. The spleen is an easy organ to access, and with all the periarterial or lymphatic sheaths, there will be a lot of lymphocytes that react to that antigen that's being introduced. So we could have 10 lymphocytes, each one recognizing a different antigenic determinant or epitope on the surface of that antigen. So how do scientists isolate that? The spleen is removed from the mouth and is kind of mashed up, macerated, because remember it's just basically a bag of cells, and mixed with growth fluid. So let's say that we have 10,000 cells per milliliter. Right? If we dilute this out, we can get a ratio of just 1 cell per mil. Now that would require 10,000 mils. All right, and that we don't need to make that much fluid, so we can just take a micron, a microliter, which is 1,000th of a mil, and put 1 microliter into 10 mils, and we would have a ratio of 1 cell per mil. And so you dilute that out. The math part isn't what I'm focusing on here. But let's say that we put 1 mil of growth fluid in here and put 1 microliter into each of those, or whatever dilution factor we need, and we would end up with 1 lymphocyte in each of our wells. Those lymphocytes would clone themselves, they would undergo mitosis, and make the same copy over and over and over. So whatever antibody they're making would be the same antibody. And so you might get some wells that have 2, you might get some wells that don't have any. But then you would run tests on these, and all of these lymphocytes would be making the same antibody. In other words, they would all be recognizing the same epitope. And that's a monoclonal antibody. So if you buy a vial of polyclonal antibodies, that means that multiple lymphocytes were making those antibodies, because one lymphocyte is going to make one recognition site antibody. Okay? So I just used that. We're not going to really talk about lymphocyte antibodies until tomorrow, but I wanted to use that to illustrate that an antigen has multiple surfaces that are actually the recognizable structure.
What's your called
The antigenic determinant, or epitope. Okay? Also on page 64, you have the word hapten. And a hapten is, sometimes our antigens are too small. They're not antigenic. They're too small to cause the body to react to them, and we sometimes want the body to do so, either through protection for diseases or for research. And so they are molecularly bound, chemically bound to a larger molecule, and the complex then becomes antigenic. So that larger molecule that they're bound to is called a hapten. Okay? By themselves, they would be too small to do so. Now we talked about cells in the lymphatic system, so we're going to come back and look at those cells again. And we have T lymphocytes. So as such, without using monoclonal antibodies, just looking at a T lymphocyte, we wouldn't be able to recognize it from an inactivated B lymphocyte. Its surface is still going to look the same. You'll notice that here it is directly attaching to a tumor cell. And when we look at the function of T killer cells and T helper cells, they have a direct connection. Once it kills a cell, it can separate and go on and kill another cell. All right? So if you look at this as far as our armed forces, this is hand-to-hand combat. The B lymphocytes that are making antibodies are more like ships sending off surface-to-air missiles. And they're quite a few miles away from where the action is occurring. We have a variety of types of T cells. So today we're going to be talking about the T helper cell and the T killer cell or cytotoxic cell. And then we also have suppressor cells that suppress. We talked about this happening with pregnancy, where the implantation site of fetal tissue, its immune response is suppressed, so it's not as sensitive to the father's proteins that would be on the fetal cells, and the mother's uterine tissue does not react to that. So this is a B lymphocyte. You can see in the upper right-hand corner, it looks very similar to the T lymphocyte. But once it becomes activated and actually starts making antibodies, it has additional cytoplasm. Remember, our lymphocytes in the blood had just a thin rim of cytoplasm? Once it starts making antibodies, it needs Golgi and ribosomal ER. So the cytoplasm grows, the nucleus, unless we cut it in cross-section here in a longitudinal section, the nucleus is typically eccentric. It's off to one side. And you can often see a light area here where the Golgi is located. The Golgi doesn't have ribosomes on it, so it stays a little bit lighter. And this is unusual, actually, to see plasma cells with red blood cells. Plasma cells typically are activated in connective tissue, where we don't have red blood cells. And then B lymphocytes are one of our three types of antigen-presenting cells. Well, that's one of the processes we're going to talk about today. And the other two, I don't have the word dendritic cell on your paper, but you can add that. So macrophages, B lymphocytes, macrophages. Here's a macrophage here and plasma cell. So here's a lymphocyte. Notice how it doesn't have very much cytoplasm apparent. The plasma cell does. So that's an activated B lymphocyte. And here's the macrophage, activated monocyte. So the macrophages and the dendritic cells, along with the B lymphocyte, function as antigen-presenting cells. So APC. So today we're going to focus on the role of T helper cells and how they activate T killer cells. And then we'll spend just a few minutes looking on how they activate B cells, and then tomorrow we'll look at that again and focus on the types of antibodies and types of immunities that B cells provide. What they have in common is that both are activated by exposure to an antigen. And the resulting behavior is that immune response. So here is a macrophage or dendritic cell presenting a small antigenic fragment to a T helper cell. T helper cells then function to activate either the T killer or the B lymphocyte. So we need the T helper cells for either of those pathways to work. The HIV virus gets into the T helper cells and kills the T helper cells. So someone who has active AIDS will have a very low T helper cell count, like 2 or 3, or even none at all. Right? Without that, there's the can succumb to a great variety of infections because they don't have either of those specific forms of immune system. So there is two different types of molecules that are present on the surface of cells that bind to these antigens. Fragments of a viral wall, parts of a tumor cell, parts of a bacterial wall, and they're known by 2 names. I typically use the term major histocompatibility, which means tissue compatibility. Oh, I wrote that once. Complex. Another abbreviation, MHC. All right. These are classified as MHC1 or MHC2, and we'll talk about the differences between them. In micro, you might have used this term instead of MHC. It refers to the same protein, surface protein, but it's human leukocyte antigen.
Those are the same thing?
Yes, but just in the human. So it's the same thing, HLA. So when someone's having a transplant and they want to match the surface markers, they're talking about HLAs. And there's 2 groups of, there's actually a whole bunch of subsets, but there are 2 groups of those as well. And when I copied this image, it got rid of my white background. But anyway, there are genes that produce these on chromosome 6, and they'll produce the MHC or HLA complexes or proteins. I keep forgetting I'm going to bring in this book one of these days, but about 15 or 20 years ago, it may have been Robin Cook, it may have been an author similar to him, wrote a science fiction drama type book in which genetic engineering was used and sold as a health insurance policy to very wealthy individuals. For several $million, you could purchase this health insurance policy, and they would take cells from your body, somatic cells, isolate the nucleus from that cell, and take the chromosome 6 segment for the HLA or MHC complex, and using genetic engineering, insert it into chromosome 6 of a type of primate in Africa known as a bonobo, similar to chimpanzees and gorillas. And the embryo, not a grown one, but an embryo of the bonobo, implant that in a female bonobo's uterus, and when that embryo developed into bonobo, all of its self-markers would have your self-marking proteins, your MHCs or your HLAs. So if perchance you happen to need a heart transplant, all right, or a lung transplant, you could call in your marker, your $1 million that you paid for that bonobo. They were raised on an island in the middle of a river, and they would capture it, take it to surgery, take out its heart and fly it to wherever you were. And then when the heart transplant occurred, you wouldn't need to take immunosuppressive drugs because all the cells, muscle cells and connective tissue cells in that heart would be accepted as your heart. It would have the same marking proteins that your heart does. And if you were scared that you had a lot of genetic problems in your body, you could purchase a bonobo for its lungs. Obviously, you couldn't use the same bonobo 5 years later for its heart if you already took its lungs. You might have to spend several $million to get a family of them for any organ that you might need. Well, of course, that was a nice story. But to put the drama in the story, apparently they took too much of chromosome 6, not just the HLA or MHC alleles, and the bonobos, one day the carekeepers, scientists looked across the river to the island and they saw smoke rising from the treetops. Bonobos had discovered fire. And they rapidly were evolving into humans. And were developing bonobo rights. And started rebelling against being harvested for their organs. Yeah, it's a published novel. I don't know if it was made into a movie or not. But anyway, it's based on this scientific, medical aspect of our self-marking proteins.
Have you seen that movie? It must be from the book.
I read the book.
It's called
Yeah.
The Island, but it's
There you go, yeah.
it's cloned people. It's not like
Oh,
Yeah.
Yeah.
with Val
Random.
Kilmer?
Ewan McGregor, I think, is in it.
Yeah.
The new one's
No, I haven't seen the movie.
You should watch it in class.
Class assignment, huh?
Yeah, Michael Bay movies sure are.
Extra credit.
So these, well, look at how they work, but these molecules are the self-marking proteins, okay? And there's two different types that we're going to look at now. So I'm laying the groundwork for some terms and fundamental concepts that we need to understand T helper and T killer cell activation. So that video's not working, so let's move to the next one. So there are two classes. I'm just going to use the term MHC, okay? I just want you to know what HLA stands for and that the terms are interchangeable. There are two classes of major histocompatibility complex molecules, class 1 and class 2. All of the cells in your body that have a nucleus demonstrate MHC1 on the cell surface. All right, so back to this earlier image that I had skipped over because it was in the wrong place in my slide. All the cells in your body are going to have these self-marker proteins, except for red blood cells because they don't have a nucleus. So how do they get there? It's how your body knows that you're you. Okay? So how do they get there? They are endogenous peptides. When we talk about MHC class 2, we're going to use the term exogenous. So endogenous means from within. Exogenous means from outside. So how does your body make proteins? Transcription of the DNA into messenger RNA. Messenger RNA is translated using the transfer RNA. And if their proteins are going to be used within the cell itself, that happens in the cytoplasm. And if they're proteins that are going to be sent to the mitochondria or sent outside the cell like antibodies, we use Golgi apparatus and ribosomal ER. Okay? So this is representing the plasma membrane surrounding the cell, surface membrane, and this is representing the endoplasmic reticulum. The proteins are the endogenous peptides, means the cell makes the proteins. The cell has the DNA and it's transcribing it and making the actual proteins. And then those proteins are inserted into the endoplasmic reticulum. I'm going to diagram this on the board for you in just a moment. So here's another larger image. Now those proteins can be huge. It can be, several hundred amino acids long. So we have our cell membrane. So this is the cell surface membrane. The cells, ribosomes, are making a protein which is going to be functional within the cell. This is our endogenous peptide, okay? Because the cell synthesizes it with your DNA. But it's too large to bind to the MHC molecule. So it passes through a structure in the cell that clips it into smaller pieces. That's what's happening here. Your cell doesn't have scissors, they're just using that as an illustration to show that clipping is going on. And then within the endoplasmic reticulum, these would be the ribosomes of ribosomal endoplasmic reticulum. There are channels that allow these peptides to pass through, and this is known as TAP, the transport something protein. I don't remember what the A stands for. And these small peptides then are inserted into the endoplasmic reticulum, where normally they wouldn't, if they're going to be used within the cell. So that's what's happening here. They may or may not get clipped and cleaved into smaller components within the ER. And then they will attach to a membrane protein made by the cell. And this is the MHC class 1. This passes through the Golgi, is pinched off, and there is a segment of it so it knows it needs to go to the surface. So it ends up fusing with the surface membrane. And now we have on the surface of this cell an MHC class 1 molecule with an endogenous peptide being presented on its surface. And as we'll see in a few moments, the T cells learn, quote unquote, they don't have brains obviously, but they learn in the thymus to recognize your own MHC1 molecules and to not react to your own self-antigens. So again, all your cells that have a nucleus will have these MHC1 molecules on them presenting fragments of the peptides made by that cell. And in the thymus, your T cells are, if they react to those endogenous antigens that your own cells make, they have to kill themselves. They have to commit suicide.
Are we supposed to know what's happening? I'm sorry.
No, they're just the channel by which these get into the ER. Because we don't want our body having an immune reaction to the markers on our own cells. So we want to get rid of those T cells that have that reaction. So again, these display endogenous peptides, and they're found on all nucleated cells.
So on the diagram that you gave us, is
Uh-huh.
that the self-antigen?
Yes.
The endogenous antigen?
Uh-huh.
And then what's the foreign antigen?
All right, so how would we get a foreign antigen onto an MHC? That was a nice segue, thank you. How do we get a foreign antigen onto an MHC1 molecule when it's supposed to be self? I'm not erasing it. When it's supposed to be self. All right, let's say that we have a virus-infected cell. When a virus infects a cell, it co-opts the cell's nucleus to synthesize new viral proteins, to make new viral capsules, and then those are shed from the cell. The cell doesn't know the difference, that those are the virus genetic components that it's using to make new proteins. And so those viral proteins are treated just as if they were the cell's own proteins made from the cell's own DNA. So it binds to an MHC1 antigen, but it's really a foreign antigen rather than being the cell's own proteins. All right? Another source would be a tumor cell. Spelled both ways, which way do I like this? So in cancer, we know that the DNA is mutated. The cell loses its natural inhibition to stop dividing, and it also makes new proteins that it wouldn't normally make, that the T cells of the body wouldn't have been exposed to in childhood, and taught that these are normal self-antigens. So when it sees them later, they're not normal self-antigens. They would be foreign antigens, even though they were made by the body cell. And then we have foreign antigens on a transplant cell. So a transplanted organ would be foreign. Also, the MHC would be foreign, and so it's a little unclear how the T cells will still interact with it because it's not a self-MHC1 molecule, but they will react with those. So these are the three major types of cells that the body responds to that have the wrong antigen on an MHC1 molecule. So the MHC1 molecule specifically interacts with and binds to a T killer cell. And we had T helper cells and T killer cells. So the cell type that reacts and responds to an MHC1 molecule, and I got a little bit more room down here, so let me redraw another one. So the cell that responds to this is going to be a T killer cell, or T cytotoxic cell. To respond to it, it has a receptor, and we'll look at this process in more detail, that recognizes both the antigen and the MHC class 1 molecule. So that's what's going on here. All right, both the antigen and the MHC class 1 molecule. And killer cells always stabilize its reaction via a unique molecule known as a CD8 molecule. So T killer cells are also known as CD8 cells. Yes, Connie?
Are there several MHC molecules on a single?
Oh yeah, the entire surface, yeah, you're going to have a lot of them. And they're going to all have different antigens, because the cell makes lots of different proteins, and different parts of them will be on different MHC molecules.
Where the T killers coming in, is that a foreign antigen?
Yes. All right, so we'll come back to this interaction here when we talk about how we activate T helper and T killer cells. But I just wanted to emphasize that MHC1 molecules, and let me emphasize that by adding it up here. So we're going to associate T cells, or you can focus different. MHC1 have endogenous peptides. Are found on all nucleated cells. Interact with T killer cells. The interaction is stabilized by, I'll just put CD8 over here. Okay. Oh. Secret the file, secret the email, I'm trying to get your phone number. I think you got your wallet and find out. She has it, yeah. Sarah, come out.
My wallet is in my backpack.
Double check, we'll just go to science office, it's like a long, oval, brown and yellow, gold.
The
Science office is
Skills Center?
440. Yeah, not the Science Skills Center, but go through the courtyard.
Okay,
Her name is Chris, she said she'd have it in her office.
thank you.
Uh-huh. All right, so now let's look at MHC2. Those are the two types of major histocompatibility complexes.